Good afternoon. My name is Allison Clune, and I'd like to welcome you to the Indoor Air Quality Science webinar series hosted by the U.S. EPA's Indoor Environments Division. Today's webinar, Investigating Indoor Chemistry, will be presented by Dr. Ray Wells. Dr. Wells began his research career in chemistry as an undergraduate at the Georgia Institute of Technology, investigating hydroxyl radical and nitrate radical kinetics. He received a PhD in physical inorganic chemistry from Northwestern University in Evanston, Illinois, exploring the gas phase kinetics of organometallic compounds. Afterwards, Dr. Wells worked for the U.S. Air Force for 7 years, assessing the atmospheric chemistry of industrial solvents. In 1999, he joined NIOSH, the National Institute for Occupational Safety and Health, in Morgantown, West Virginia. Since 2001, Ray has investigated the gas phase, surface phase, and particle phase chemistry of terpene oxidation present in indoor environments and worked to develop new sampling technologies. He began his current role as branch chief of the Exposure Assessment Branch in the Health Effects Laboratory Division of NIOSH in September 2019. Thank you so much, Ray, for being here with us today. This webinar is being recorded and will be made available on the EPA website at a later date. Please note this presentation reflects the opinions of the speaker and not necessarily those of the U.S. EPA. If you have a question during today's presentation, you can type it in the chat panel on the right-hand side of your screen at any time. Questions will be read to the speaker for discussion at the end of the presentation. If you have any technical questions or comments, you can also submit those to the chat box. We look forward to hearing from you. So, Ray, thank you again for speaking today, and I'll hand it over to you to begin the presentation.
Well, good afternoon, everyone, and thank you very much, Allison. And I'd also like to thank the EPA for the opportunity to give this presentation. As Allison said, I'm from the National Institute for Occupational Safety and Health in Morgantown, West Virginia, and today we'll talk about investigating indoor chemistry. Just to give you a little bit of a background of where NIOSH is located, NIOSH as an institute has operations all around the United States. The home office is in Washington, D.C., but Morgantown is just south of Pittsburgh, Morgantown, West Virginia, and this is a picture of our building, and we have 4 divisions that are located in Morgantown. The Division of Safety Research, which investigates occupational safety in construction sites or mechanical engineering. The Respiratory Health Division, which investigates respiratory health exposures, field sampling, and surveillance. The Health Effects Laboratory Division, the division that I'm in, does fundamental research all the way from engineering of sampling devices all the way to fundamental biological processes. And part of the National Personal Protective Technology Laboratory, which one of their functions is the certification of respirators, also does fundamental research on exposure both here and in Pennsylvania. So the outline for my talk is shown in this sort of pictorial representation here. So at the top, we have several motivational elements that I'd like to share with you that have highlighted our interest in trying to understand or investigate indoor chemistry. That motivation leads us into the reaction chemistry bubble, if you will, where we'll talk about gas phase chemistry, particle phase chemistry, and surface phase chemistry. And note the importance that the arrows between those boxes are double-sided arrows, meaning that all of these 3 things work together, even though we'll talk about them separately. They're all happening simultaneously and can influence one another. These concepts of the chemistry of the indoor environment will then be placed into techniques or experiments where we're going to talk about trying to understand health effects, or at least peel back some of that interest. And then that will give us new insight into other experiments, as well as new research directions. So the start of the motivation section of the presentation really began earlier, but it was sort of formalized in 2001 with the National Human Activity Pattern Survey, where over 9,000 people kept a dialogue or a catalog of what they, how they spent their time, and they could clearly show that they spent 86% of their time in the indoor environment. And so this was a very interesting finding that helped us realize that the indoor environment exposures were important. And to take this a step further, Rich Corsi a few years ago developed this series of numbers here. He didn't call it the Corsi code, but it is called the Corsi code. And it is represented, these series of numbers mean this thing. The average lifespan is 79 years. 69 of those years we spent indoors. 54 of those years we spent in our home. 26 of those years are in bed or sleeping. 4.3 years in vehicles, and only 6 years of our life are spent outside. So again, this helps to bring sort of some concreteness to the 90% of our time spent in the indoor environment. Other motivational elements that I'd like to point out is that for many chemicals, concentrations are higher in the indoor environment than outdoors. And this is in part because of the enclosed space and sometimes the lower ventilation as compared to the outdoor environment, but also our use of consumer products, bringing different materials into the indoor environment and using these products. Cleaning has been associated with an increase of asthma and respiratory symptoms, certainly in the occupational sense, and this can cost up to $56 billion in direct and indirect costs, such as lost days of work and lost productivity. And there's an entire research effort to try to understand these costs and put them in perspective. Even though the costs are large, a direct cause and effect is still not known. To further highlight the importance of the indoor environment, I found this report from the Bureau of Labor Statistics, which was actually just released this September, but it shows that from the years 2019 to 2029, there will be a significant increase in healthcare support occupations. And this is important for what we're going to talk about today for a couple of reasons. One, healthcare facilities and healthcare occupations are typically in very cleaned or multiply cleaned environments, and they're also typically an indoor environment. So we're likely to have more workers in the indoor environment as the future moves forward. So our interest is to characterize the indoor environment by looking at the chemistry, but taking it, as I said earlier, a little step further to understand sort of the occupant exposures, and then also understand how these exposures could contribute to health effects. As a team, we have focused on sort of what I consider the three parts of this issue of indoor chemistry, if you will. So we'll talk specifically about chemistry and mechanisms, which we obviously those are important. But to translate the chemistry and mechanisms into a field or a real world environment, we need detection methods that are easy to apply, inexpensive to use, but still give us that detailed information. And I'll talk about a couple of methods that we've used. Our ultimate hope, again, because of the goal of the National Institute for Occupational Safety and Health to protect workers, there are several industrial hygienists in the institute that go into the field to sample workplaces. And this, we hope, is a home for the translation of our chemistry research into new and viable detection methods. But the other corner piece is understanding the toxicology, and that's the word I'm going to use for health effects. What biologically happens? Why do these health effects occur, and can we reduce them or eliminate them? So this helps us to understand a definition of indoor air quality, even though indoor air quality is not clearly defined per se, but if we think about the health effect species and the biological interface, we can certainly improve the case where we reduce the number of illnesses. So I'll talk about the chemistry research. We'll talk a little bit about ozone and its transport from outdoors to indoors. Terpenes are going to be our key class of compounds. Terpenes are natural oils from plants, but they're present in many of our consumer products. And we'll look, as I said earlier, at the gas phase, particle phase, and surface phase chemistries. So the starters of all of these chemical processes that can occur indoors are typically these three species. The main reactant or oxidant is ozone. Ozone is formed outdoors from air pollution, but because buildings interact with the outdoor environment, they breathe, they pull outdoor air inside, ozone is transported indoors. There are some losses as ozone is moved indoors, but typically the concentrations of ozone indoors can be between 20 and 50 parts per billion. Ozone mainly reacts with alkenes, or molecules that have carbon-carbon double bond, represented by the C double bond CR structure there. The products are oxidized compounds, OH radicals, which I'll talk about in a minute, but they are very important, and secondary organic aerosols, also another important class of product. The OH radical is also present outdoors, but it is so reactive that it's not transported indoors. It is actually formed indoors, and there have been recent measurements to show that the OH radical concentrations in indoor environments can at times be as high as they are outdoors. As I mentioned earlier, the ozone reaction with alkenes can form the OH radical. The OH radicals are very reactive, as I said earlier, but the key thing that they do is they abstract hydrogens from carbon-hydrogen groups to make radicals, other radicals, and they can also react with alkenes like ozone does. They bridge across the alkene group, again, breaking that open, leading to other radical sites. The products are carbonyls, carboxylic acids, or other oxidized compounds and secondary organic aerosols. One of the radicals that typically is in the nighttime environment outdoors is also highly reactive like the OH radical, and it's also formed indoors. Ozone can react with NO2, which is a combustion pollutant, to form the NO3 radical. And again, its concentrations are are 10 to the minus 3 parts per billion. And like the OH radical, the NO3 radical can react with CH groups as well as alkenes in a similar fashion. Its main products are nitric acid, other oxidized compounds, and secondary organic aerosols. So we've talked about terpenes, and I wanted to show you some of the terpenes that are present in many of the products that we use, both as cleaners or as fragrances. And you can see the structures here, as well as their olfactory notes and quotes underneath the structure of the compound. You'll notice also, too, that some of these compounds have 2 carbon-carbon double bonds, which are shown by the secondary line on the structure. 4 of them are ringed compounds, which means that when the carbon-carbon double bond is broken open, you have the whole molecule still stays together, but it's just broken apart now. Dihydromercenol, which is the lavender scent, is a sort of a straight chain hydrocarbon with an alcohol on one end, and that carbon-carbon double bond on the very other end of the molecule. So our basic reaction scheme that I want to show you is the ozonolysis reaction. So ozone can add to the carbon-carbon double bond, shown here as a sort of a basic structure, and it makes what's called a primary ozonide, where all 3 oxygen atoms bridge across from one carbon to the other on the alkene. This leads to the formation of a highly energetic Krigge radical, and you notice that it's a biradical. There's a radical site on the carbon and on the oxygen at the end. And the carbonyl that's formed from this breakup goes on to be into the gas phase. But what's interesting is the Krigge radical, that oxygen on the end wants a hydrogen very badly, and so it'll reach anywhere and pull a hydrogen off of the molecule, creating another carbon-carbon double bond, but then it makes this group that can dissociate into the OH radical, which we talked about earlier, and another alkoxyl radical. So overall, globally, we're looking at carbon-carbon double bonds reacting with ozone, OH, or NO3 to make this whole suite of different types of structures of products. So there's a lot of transformation that can occur in the indoor environment. So we'll look at the gas phase chemistry first, and we'll talk about some of the investigation tools that we use. We use a Teflon chamber, which is an 80 or 100 liter chamber that we can make in-house that has ports on it that we can either introduce or withdraw chemicals from. It's typically housed in a wooden box, sometimes with lights to simulate radiation of some sort. But then we pull the contents out of the chamber and process them in ways that we'll talk about in a minute. And our typical analysis tool is the gas chromatograph mass spectrometer. And the advantage here is the gas chromatograph allows us to take a mixed sample, and through chromatography, we can separate that mixed sample into individual components typically. And the mass spectrometer helps us identify the products that are formed because each of the chemicals has a specific mass signature as it's broken up with an electron beam. So we'll talk about the ozone reaction first. And the reaction that we're going to talk about is alpha terpineol plus ozone. And so if you remember, ozone is going to add to that carbon-carbon double bond in the ring. So the way we do this experiment is we take our Teflon chamber and we load it with a very high concentration of alpha terpineol, 500 ppb or up to 2 ppm. And so into that high concentration alpha terpineol chamber, we inject a small amount of ozone and we monitor the ozone loss over time. And the ozone loss over time gives us an indication of this K prime, which is shown here on the y-axis, versus the concentration of alpha terpineol on the x-axis. And we get a linear plot, and the slope of that line gives us the rate constant for alpha terpineol plus ozone, which we can see here. Now we're also going to investigate the OH radical reaction, and again, we do this a little bit differently, still using the Teflon chamber, but in that chamber we load alpha terpineol and the OH radical precursor and something that's called a reference compound. So in this case, it's either isoprene or 2,5-dimethylfuran. And then the technique that we use is called the relative rate technique. And isoprene and 2,5-dimethylfuran have very well-known OH rate constants. And to try to figure out the unknown OH rate constant, the rate constant we're interested in, alpha terpineol plus OH, we can initiate an OH reaction and monitor the loss of alpha terpineol and the loss of either isoprene or 2,5-dimethylfuran as the reaction proceeds. And we end up with a plot that looks like this. And it's a linear plot. The slope of this line gives us the rate constant of alpha terpineol plus OH. The advantage of using two separate reference compounds gives us a much better confidence in the number that we've measured, simply because of the difference between the isoprene OH rate constant and the 2,5-dimethylfuran OH rate constant, but we're still getting a linear plot. And the rate constant, I should note here, is quite fast. This 2 × 10⁻⁻10 is almost gas kinetic, meaning that as soon as OH runs into an alpha terpineol molecule, it reacts. So for the NO3 reaction, again, we're using the relative rate technique, and you can see our reference compounds are 2 carrying and 3 carrying. Again, a similar type of data, a linear plot that shows both of the reference compounds on it. And again, the rate constant of 16 times 10 to the minus 12 is also a very fast rate constant. Oh, sorry. And so to try to put, and again, we've done several rate constant measurements, but I just want to give you an idea of how we did those experiments. And so now we can put some of this information into context. So if we look at the top, where we say typical air exchange is between 0.6 and 8 per hour, which is the number of air changes in a building, we can look at that as a loss rate. And so the question is does terpene oxidation chemistry compete effectively with the loss that would occur from air exchange? And we can show that in some cases it does. So if we look down on the left side under the organics column, we can see that alkenes, all alkenes, have an ozone rate loss of about 0.7 per hour and the nitrate radical rate loss of 0.56 per hour, which is sort of slow. But if we go down to the bottom of the table and look at the terpenes, we can see just average, the terpenes react much, much faster, almost 10 times faster than the average of the alkene reactions. And both for ozone and NO3 radicals seem to be fast enough so that the chemistry, we have to think about the chemistry occurring in the indoor environment. If we look one row up at the dicarbonyls, unsaturated carbonyls, we think that these classes of compounds are important to try to understand health effects. And the other thing that's important to notice too that is that its reactions with ozone or the NO3 radical are actually quite low. So that means that once they're formed in the indoor environment, their only loss is actually through air exchange and not necessarily through reaction. So that's an important thing to think about as compounds can start to build up in the indoor environment. So while there is chemistry that we've investigated, all of those rate constants can go into larger models that look at the fundamental chemistry. And in a recent publication by Nick Carslaw at the University of York, she used a model to look at different types of compounds that are formed from the limonene plus ozone reaction. The limonene plus ozone is on the left side of the plot here. But I just want to focus right now on the carbonyls, and you can see that the carbonyl compounds are modeled to be formed and are the gas phase precursors for secondary organic aerosols. So again, here's another reaction schematic, and this is terpinolene this time, but I just wanted to show you the type of products that can be formed. So if ozone attacks the top carbon-carbon double bond, the one that's external to the ring, we get this primary ozonide, and here's our friend, the OH radical is formed, and then we make the carbonyl compound. Now, if ozone reacts to the carbon-carbon double bond that's already in the ring, then we make this primary ozonide, but notice that we've got not just a single carbonyl compound, but a dicarbonyl compound. And just to give you some foresight about the health effect studies that we're thinking about, is the multiply oxygenated species have the tendency or the thought that they could impact health effects. So here we have a dicarbonyl compound that's formed, and of course we also make the OH radical from this reaction as well. One of the challenges with the carbonyl compounds is that they can be difficult to manipulate and inject into gas chromatograph systems. The challenge there, they can get lost on the injector or in on the column. And so one of the things that has been done is to derivatize these oxygenated species. And so the basic reaction is shown at the top, where we have a carbon double bonded oxygen plus D, which is our derivatization compound, to make a carbon double bonded to the D complex. The advantage here is that we have a unique marker to show that yes, this this was a carbonyl compound before we converted it to the derivatized compound. And the other advantage when we look at the mass spectral data is that the structure, the fundamental structure of the molecule has been retained, which is very important for trying to understand reaction mechanisms like the slide I just showed you. So the two types of derivatization compounds that we use are PFBHA, shown on the left, and its structure, the perfluorinated benzene ring. And it should be noted that for each carbonyl group that gets derivatized, it adds an additional 195 mass units to the fundamental molecular weight of the compound. And we also took advantage of the same chemistry, but we used a smaller molecule called, we call it T-box, and again, it only adds 71 mass units. There are some advantages here. So if we look at the sentence below, a tricarbonyl will add 585 mass units with PFBHA. So if your mass spec is limited in its mass range, you're already hitting a you're almost at the end just with PFBHA alone. So the advantage of using a smaller derivatization agent is shown here, but only 213 mass units added with T-box. So the way we do these experiments are we load the chamber with the terpene of interest and add the oxidant of interest, ozone OH radical or NO3 radicals, initiate a reaction, and then collects that sample into, and what we're using here is water. We're pulling at 4 liters a minute into 25 mils of water. The advantage of water is, if you remember, we're trying to think about techniques that can be easily used in the field, and so water is a relatively safe compound to take into the field. And we've also developed a little technique to be able to add derivatization agent in the field as well, in a controlled way. And so once we add the derivatization agent to the collected sample, we can heat that for a couple of hours at 70 degrees C to sort of facilitate the reaction of the derivatization agent with the carbonyl compounds, and then we can use a little bit of toluene to extract the derivatized compounds into a thin toluene layer, so the concentration is high enough for detection, and then we can inject some of that toluene layer onto the GC mass spec system. And so some of the data that we would see is something like this. So again, we've got terpinolene reacting with ozone, and you remember we make the ringed carbonyl compound and the dicarbonyl species. So the green data is just terpinolene alone, where we load terpinolene into the chamber, pull a sample, and then add derivatization agent. And the red data is terpinolene after it's reacted with 75 ppb of ozone, again pulling that the contents of the chamber into water and then adding the derivatization agent. You can see that at least in this data, we see about 4 reaction products: methylglyoxal on the left, the ringed carbonyl next to it, and then down at the end, around 27.5 minutes, you see that we see the dicarbonyl species that we mentioned earlier. But notice too that we see a tricarbonyl species, and again, the more oxygenated a compound that might be important for health effects. So we were able to detect the tricarbonyl species using the T-box derivatization agent. So doing things in the lab is interesting, but we also need to try to do field sampling and see if we can address the other component of our work, is being able to translate our laboratory discoveries or efforts into the field. And so we collected a 2-hour, 480-liter sample from an office building atrium. We used the T-box derivatization agent, and you can see that we can detect, if we start looking on the left around 15 minutes, glyoxal, methylglyoxal, and then a compound 4-oxopentanal, which we have observed as a reaction, dicarbonyl reaction product from many of the terpene species, as well as from squalene, which again, I'll talk about that in a minute. And so this could be an important dicarbonyl compound present that typically is not measured routinely. And then down at the end, close to 30 minutes, we've got a di or a tricarbonyl species. So revisiting the model and the figure that I showed earlier, not only are carbonyls expected, but organic nitrates are expected to be formed in the indoor environment as well. And so the organic nitrates can make a significant contribution to secondary organic aerosols. So the way we do these experiments is to take our chamber with alpha pinene and react it with ozone. And the reason that we chose alpha pinene is it makes typically the yield, the OH radical yield, is fairly high. And so the OH radical is formed from this reaction. We take advantage of that by adding cyclohexane to the chamber as well. Cyclohexane will react with the OH radical. The OH radical abstracts a hydrogen to make water, and then the oxygen that's present in the air will react with that radical site to make this peroxyl radical, which can react with NO and go two different directions, one to make NO2 and an alkoxyl radical, but the other reaction that we're interested in is the formation of cyclohexyl nitrate. And because this is a dynamic system, it's not a linear process either. When we vary ozone or NO, you can see that we get this sort of reaction shape. And this is important to try to understand the multiple processes that can go on to lead to the formation of nitrate species. So investigating particle phase chemistry, we can look at the overall process where a volatile organic compound, a terpene or some other volatile organic compound, can react with an oxidant. And the key here is that the reaction product, either the primary or the secondary reaction products, can ultimately be low vapor pressure compounds. And so these compounds would prefer to go to surfaces, and they have several routes, but two of the routes that we think are important is they can condense with other low vapor pressure organic compounds to form secondary organic aerosols, or there is particulate matter that's almost always present indoors. These low vapor pressure compounds can partition to particles, and that would be transported indoors, and again leading to the formation of secondary organic aerosols. And in a recent publication from Drexel University, they investigated the composition of particulate matter. And this is important to try to get an idea, again, of the interaction between the outdoor environment and the indoor environment. And so they had a very special instrument, mass spectral instrument, that could collect particulate matter and characterize it. And the pie chart here shows the characterization of the particulate matter that's outdoors. And they measured about 11 micrograms per meter cubed, I think was in the article. And again, you can see the different types of species. And the two we want to focus on today are the gray pie piece, which is the hydrocarbon organic aerosol, and the NO3, which is the blue piece, the nitrate aerosol. And so in the outdoor environment, again, they observed this sort of splitting of species. But when they sampled indoors, again, these particles went from the outdoors to the indoor environment. There was some mass that was lost, hence this is why the pie shape's a little smaller. So it's down to 6 micrograms per meter cubed. But look what happened to the hydrocarbon, the HOA portion. It went up significantly to 19%, and the nitrate aerosol decreased significantly to about 4%. And so again, this is a sort of an important highlight of changes that can happen from the outdoors to the indoors, as well as the importance of the interaction between the outdoor environment and the indoor environment through ventilation. So we used a denuder filter apparatus to look at some of the contents of our chamber from the particulate matter. And again, we can initiate a reaction in the Teflon chamber, but then pull the contents of the chamber through a cyclone, which can pull out the 2.5 micron particles, But then the gas phase species goes through what's called a denuder. And this is a series of concentric glass tubes that are coated with adsorbent. And the gas phase species will diffuse out of the gas phase onto these surfaces, and they're trapped there or absorbed there. The particulate matter, because of its momentum, can travel through the glass tube and then impact on the filter. So this allows us the ability to look at both gas phase species and particulate phase species from the same sample. And very quickly, I just wanted to show you what we observed from alpha pinene plus ozone. Again, the main product here is shown on the right. Its structure is called its pinenaldehyde. It's a dicarbonyl species. And the data that you see here, the red line, is just simply an air sample that was run through the system, and the derivatization technique was added to that sample. But the orange data is the pinenaldehyde concentration that we observed in the gas phase. But importantly, the green data, the filter data, shows that pinenaldehyde is also present in the particulate matter. And again, you know, it's not either or. We can have gas phase species present in both the in the gas phase as well as in the particulate phase. In order to try to understand the impact of particulate matter as well as the influence that mixtures might have, again, we started with alpha pinene plus ozone, and we reacted that in the presence or absence of amine, and the structure of this amine is shown above, and it's ethanolamine. And ethanolamine is a compound that can be present in either cleaning formulations or surface finishes. And we were curious to see what its impact on secondary organic aerosol was. So when we did this reaction, we get these plots here. And so on the bottom is time, and on the y-axis is the summed mass. So we take the contents of the chamber and react it and flow it through a particle size analyzer, and we look at the summed mass across all particle sizes, and we get these plots that you see here. So interestingly, when there's no ethanolamine, we get the orange data. When we add a little bit of ethanolamine, we see an increase in the summed mass, suggesting that the ethanolamine reacts with some of the products that are formed from alpha pinene ozonolysis to lead to low vapor pressure products that can end up in the particulate matter. But the black data is very interesting because even though we simply doubled the ethanolamine concentration, we more than doubled the level of the summed mass that we observed. And so what we would have expected if this was a linear process would have been a level at the green dashed line, and that is not what we observed. We observed a much higher increase. So our interest in surface phase chemistry is important because the surface to volume ratio value of the indoor environment is much, much higher than outdoors. We have walls, we have floors, we have carpets, we have furnishings. All of these surfaces add up and can contribute to the chemistry that can occur indoors. And so a few years ago, we were able to construct a system that has a feedback loop into it, where we can deliver oxidants onto a surface of interest and collect what comes off of that surface over a long period of time. So we can simulate surface chemistry over several days and look at the products that are formed in a timed fashion. And so if you remember, we talked about dihydromercenol earlier, and I did highlight that it was more or less a straight chain compound with a carbon-carbon double bond at the end. And so when we did the gas phase experiments, we thought that because of the carbon-carbon double bond, we would see a reaction of dihydromercenol, and we would observe products. Well, the rate constant for ozone plus dihydromercenol was very, very, very slow, and it was surprisingly slow. And so what we think was happening is in the gas phase, the alcohol group can fold over and impact the electron density on the carbon-carbon double bond, which doesn't make it as attractive to the ozone molecule. But on the surface, when we add dihydromercenol to a surface, not only did we see reactions occur, but we also saw products that we were expecting from our gas phase experiments. And so you can see that surfaces can enhance some of these reactions because the alcohol group has been removed from, we hypothesize the alcohol group is doing the attaching to the surface, and the carbonyl is now able to be attacked by the ozone molecule. Another compound that's very interesting and important for the indoor environment is squalene. And I show its structure here. You see that it's a very long hydrocarbon with 6 carbon-carbon double bonds, so it makes it ripe for ozone reaction. It is not, has a very low vapor pressure, and it's a major component of our skin oil. So there have been some recent measurements to show that squalene is present on surfaces in the indoor environment, and that's because of the human occupant. So again, we made a solution of squalene and applied it to a surface and added ozone to that surface over a long period of time. And so here's the data that we show on the bottom. So the blue data is when we react squalene with PFBHA, and you can see that we don't really see any peaks due to carbonyl derivatization. When we did the experiment over several hours and looked at the gas phase emissions of the squalene surface ozone system, we get the red data. And you can see that there are a couple of peaks in the red data, notably 4-oxopentanal, but again, not very much as far as the emissions of carbonyl compounds go. But when we rinsed the surface, we could clearly show by the large green peaks here the formation of carbonyl species on the surface. And so not only is the surface a reservoir for chemistry, it's also a reservoir for oxidation products, which may be important also as a reserve for further chemistry. Chlorine in the form of bleach also has an impact on the indoor environment, and this is another recent publication by Jay Wong at University of York, highlighting the HOCl group, which has a green box around it here. This is the sort of the main component of bleach, and I won't go through the whole mechanism, but you can see that in their research, they were able to delineate and show the formation of all of these other compounds. So the formation of chloramines, as well as a chlorine nitrogen compound, which could be important precursors for other reactions. You can see up in the upper left the formation of the OH radical as well. One of the other things that I want to highlight is I have a question there. I say other amines question mark. So they were able to show that ammonia can react with HOCl to form NH2Cl, and then that goes on to react further. So ammonia is important because occupants, humans, emit ammonia and can contribute to the chemistry of the indoor environment, and that's temperature dependent. So the hotter it is, the more ammonia we tend to release into the indoor environment. But the other question too is other amine compounds that are present in the indoor environment, can they be part of this cycle? So while we do see some carbonyl compounds, and we can show that chemistry is occurring, as you can imagine, just like in the outdoor environment, there's a lot of unaccounted for carbon. So there's missing carbon. So if we try to think about other products that are formed, and how do we capture those? So we didn't observe a huge number of carbonyl compounds, but however, these were limited field collections. There could be other oxygenated species such as carboxylic acids, hydroperoxides, or epoxides that could be formed. And this leads us to our continuing quest for new routine detection methods that we think are needed to look for these other oxygenated species and other non-carbonyl species. So we have this body of knowledge of the chemistry that can occur, and now we're moving out of that fundamental chemistry bubble, if you will, into the other parts. And so we're going to try to translate what we've observed and try to help to understand health effects. So we'll use our indoor chemistry systems to generate species of interest, to deliver those to deliver that mixture to a biological system. And then we'll analyze the changes in the biomarkers that would be indicators of either exposure or irritation or sensitization. Now, obviously, there's some challenges here. Chemistry, for the most part, can quote unquote behave itself, but biological systems can be very complex. You have multiple cells doing multiple functions, and that complexity can cloud the data interpretation. Also, the timing of the data collection from the biological systems is important, as I'll show you in a minute. And identifying the biomarkers for analysis is still another big challenge. But before we get to that data, there's a tool that we have used for quite some time that looks at the structure activity of the compounds of interest. And so this is called the Chemical Asthma Hazard Assessment Program. And this is available on the web, and the website's shown here. And this was developed in England. And it's a structure reactivity-based idea where you can load a chemical structure into a program and hit submit, and then the structure of the compound of interest is compared across 78 known chemical asthmagens and 301 recognized occupational exposure hazards. And then there's the creation of a hazard index value. And so this value goes from 0, which is basically not an asthmagen or an issue or could cause a health effect, all the way to 1, which is an issue or something that could cause a potential health effect. So if we look at the chemistry that we've been talking about, and we look at alpha terpineol over here on the left, we can see when we load its structure into the hazard index value program, we get a value of 0.15. And this, like all the terpenes that we have observed, suggests that the terpenes themselves are not necessarily inducing health effects, but their products might be. So if we look at the table over on the right, we can see some of the other products that we've observed and have been formed, and we load those structures into the hazard assessment program, and you can see that the values are significantly greater than 0.5, and some of them are quite close to 1. So this, again, further supports the thought that a multiply oxygenated species could lead to exposure to that could lead to health effects. And these could be both in the gas phase and in the particle phase species. So, but again, this is a very easy tool to use, and it's been very helpful for. While it's not conclusive proof, it certainly helps us to identify which products we should try to think about or try to detect. So we're going to expose some cells now, and the experiment that we have done that in is shown here. So you remember I talked about 4-oxopentanal earlier. So this is a dicarbonyl species. Again, going on the logic that a multi-oxygenated species could impact health or exposure to one. So we were able to load our chamber with 4-oxopentanal and then deliver the contents of the chamber into a vitrocell system, which is an airflow system where the trumpet directs gas phase species onto a layer of cells, which are kept warm and fed on the bottom. And so once the cells are exposed for 4 hours, and then we take the cells out of the apparatus and place them in fresh media, and then proteins expressed by the cells will accumulate in the supernatant. And then we can detect those various proteins signaling some sort of exposure issue or by using ELISA. So here's the data here from our 4 OPA experiments. 4 of the pro-inflammatory mediators are shown here: IL-8, IL-6, TNF-alpha, and GM-CSF. And the concentrations of these mediators are shown on the y-axis, and the conditions, experimental conditions at certain time points, are shown on the x-axis. So you can see that these cells, when they're exposed to 4-oxopentanal, there's an increase in these pro-inflammatory mediators. Interestingly enough, if you look in the upper right, you see IL-6. Basically, at the 8-hour mark and the 24-hour mark don't indicate a response, but at the 12-hour mark, there was a significant increase in the formation of IL-6 from the cellular system. And again, this helps to point out the importance of looking at across all time windows as we try to understand the layering of signaling of cells after exposure. One thing I do want to point out is that the concentration of 4-OPA that was used, 65 parts per million, is quite high. So obviously there's still a lot of work to be done, both on the identification of pro-inflammatory mediators, as well as the delivery system, as well as the cellular system. So again, this just sort of captures the thoughts that I had. The chemical species delivery is an important component. And again, we could also do and have done systems where we initiated an ozonolysis reaction and then introduce that to the cellular system. Again, as I mentioned, the cellular system has multiple functions. And to try to think about an actual exposure, we need to really think about whole tissues. So that's multiple types of cells working together to deal with the exposure. And sometimes the mixture oxidation and aging could have an impact on how the cells are, what they're exposed to. So very briefly, I want to talk about some new research directions. I've spent the bulk of the time talking about single compounds and their reactions, but we all know that really this is a mixed system, and the reaction of mixed systems, as I'll show in a minute, is very different than the behavior of single compounds. And so if we just think about cleaners and look at their material safety data sheet, Bello et al. Did a nice job of analyzing the potential compounds that would be present, and you can see these are oxygenated species 2-butoxyethanol, alcohol ethoxylates, which are 2-butoxyethanol is a cousin. There's ethanolamine, hydroxyl alkylamine oxides, and then of course the quaternary ammonium cleaners, which are labeled or shown here on the bottom. And there are other chemicals that are present in other products. And so if we think about our mixture chemistry, here we are at our terpene plus ozone reaction again, and we're back to the Kriege radical. But the Kriege radical has some choices. In the presence of mixtures, it may create OH, or it may also be stabilized and then potentially have enough energy to react with some of these oxygenated species. And so I sort of show a generic reaction pattern here that could lead to the formation of carboxylic acids down at the bottom of the page, or another oxy radical type product, which can then go on and further react. So the question is do these products lead to secondary organic aerosol enhancement? And some of those other questions can be addressed here. I mentioned that we have recognized that mixtures could be a challenge, and so we'll take a look at the OH yields that are formed. So when we mixed at the top of the page here, we've got the structure for limonene plus alpha pinene. when we mixed those two compounds together and initiated an ozone reaction and captured the OH that was formed, we could determine how much, what the OH yield from that system was. And so you can see the percentage yield here is 74%, which is fairly high. So that almost every time ozone reacts with one of these compounds or in this mixture, you get an OH radical. So based on the known yields of limonene alone or alpha-pinene alone, we measured or determined a modeled yield of 69%. So there's a little bit of an increase when we mixed limonene and alpha-pinene together. So that's kind of important because this OH, as you can imagine, can lead to other reactions on the system. But when we look at the alpha-terpineol addition to either limonene or alpha-pinene, we can see that there's a significant reduction in OH yield. And so there's some work that needs to be done here to try to understand how alpha-terpineol, when it's in the mixture, how it can impact OH yield formation. And so again, this helps to point out an important area in trying to understand mixture chemistry. So I want to highlight a few important efforts that have happened recently. And so one of those I want to talk about is the home chem, which is the house observations of microbial and environmental chemistry. The principal investigators were Nina Vance and Delphine Farmer. And in June of 2018, they and 20 other research groups from 13 universities spent 28 days in the University of Texas at Austin at the U-Test House. And all of the instrumentation that was brought into that space was used to analyze various activities such as cleaning, cooking. They had 2 or 3 Thanksgiving dinners, I think, just to simulate activities that would be happening in the home and eventually into an indoor space. And the instrumentation really monitored all kinds of processes and chemical species. And so this was a really important test to try to begin our understanding of the indoor environment and the complex chemistry that was there. And so this was sponsored by the Sloan Foundation. So as I mentioned, there have been several enhancements to our understanding. So mass spectrometry, which is the important tool to try to look at individual chemical species, continues to get better and better time resolution, portability, as well as the ability to handle mixed samples. And again, it takes some math to sort of deconvolute those samples, but that's improving all the time. There's some other species that can be detected using cavity ring down spectroscopy. And again, this is a laser system where a laser is bounced in between 2 mirrors, and the decay of that laser light is a function of compounds that are present in that cavity. And so there are new species like chlorine and ammonia that are being detected all the time, and these are becoming much more routine instruments to use. And again, the portability on these systems are also increasing. There are new sorbents and sampling materials that are present that are being used in the field, and then also the health effects and trying to understand the interconnections as well. In summary, I'd like to point out that chemical indoor environments are chemically dynamic, and this is not a huge surprise. New tools, as I mentioned earlier, are improving characterization, and we continue to need advances in chemical mechanisms of mixtures, as I mentioned, detection methods that are both routine and specialized, as well as health effect mechanisms for understanding exposure and then adverse outcomes that can occur from those exposures. What's exciting about this is there are collaborations across all disciplines. So not only are people focusing on indoor chemistry, but the expertise that's present in the outdoor chemistry research world is collaborating in the indoor environment chemistry, as well as the inclusion of people interested in understanding health effects. And the community and its research interests are continuing to grow. There are a couple of resources that I would like to highlight that if you're interested, the Sloan Foundation website, all you have to do is say chemistry of indoor environments, search on that, and the Sloan program will take you to this page. This page is full of information on indoor chemistry. There's some educational videos, there are links to papers. It's a wonderful resource that Paula got together funding for just a few years ago and has really helped to grow the interest in the indoor environment. The home chem project that I just talked about a minute ago is at this website. Again, it is full of information and links to publications from this project, which are numerous already and have continued to expand our knowledge of indoor chemistry. The EPA Indoor website is also a very useful resource, as well as the NIOSH Indoor Air website for occupational exposures. There's some very recent publications that I have found very useful. The Royal Society of Chemistry published a book, Indoor Air Pollution, in 2019, and each chapter of the book is focused on a specific element of the indoor environment, and so that is again it's a up-to-date knowledge collection of indoor chemistry. And Bill Nazaroff and Charlie Weschler just published this year in Indoor Air article called Indoor Acids and Bases. And again, if you're interested in the chemistry, they talk about several different chemistry elements and do a very nice job. This is a very good document for just really beginning to understand the chemistry that can occur indoors and how we can utilize it. And Nina Notman just published from the Proceedings of the National Academy of Sciences, How Human Biology and Behavior Affect Indoor Air Quality. And again, this is also a very interesting article on current discoveries of occupant impacts. And again, one of those is the ammonia emissions that I mentioned earlier today. I certainly did not do this work alone, and I want to thank my colleagues from NIOSH, Jason Ham, Joel Harrison, Stephen Jackson, who's at the EPA now, Larry Lee, Crystal Forrester, Bruce Pacolay, and Stacy Anderson and her group for the health effect research collaboration. Stacy Anderson, Eva Lakomska, Jennifer Franco, and support from my division director, Don Beeshold. The collaborators, the external collaborators that we have, Michael Waring from Drexel University, Charlie Weschler from Rutgers and DTU, Nick Carslaw, Magda Cruza, and Gordon McFiggins from England, Bill Nazaroff from Berkeley, Rich Corsi now at Portland State, but he was at UT Austin, and Glenn Morrison in North Carolina. I certainly want to recognize two contributors, Mike Flemmer and Leon Butterworth, who contributed greatly to this work, but sadly are no longer with us. And that concludes my presentation.
Great, thank you so much, Ray. This is a great presentation. We have a lot of questions coming in from the audience, so I will just jump right into those. One of the first questions came from a participant asking about the use of cleaning products and disinfectants, especially with the increased use of those as a result of the current pandemic. You spoke a little bit about the chlorine radical from bleach having a role in gas phase and surface phase chemistry. Are there other important species that impact indoor chemistry during cleaning?
Well, we talked a little bit about the surface chemistry that can occur. And so there are radicals that could be formed on surfaces that could lead to the emission of other products as well. The quaternary ammonium compounds, again, are very low vapor pressure, very popular low vapor pressure cleaning compounds, which the reaction products, which people have not really investigated, could have health effects too that we need to at least be aware of or think about. And again, this opens some insight into surface chemistry.
Excellent, thank you. There were a couple of questions related to the gas phase data that you presented with ozone. One participant asked, did the studies take into consideration the time it takes for ozone to revert back to oxygen?
Once the ozone adds to the carbon-carbon double bond, it does not really convert to oxygen. It becomes a carbonyl or a carboxylic acid or some other oxygenated species. It becomes part of that new molecule. So it doesn't. Ozone does not convert back to oxygen at that point.
Okay, great, thank you. Another question related to that set of data, I think, was that the OSHA recommended limit for ozone is 0.1 ppm, I believe. And I think that the gas phase data that you presented seemed to be at much lower concentrations. Would you expect any different results at concentrations near that limit?
What, near the higher limit?
Yeah, near the higher limit.
I would not think so. I mean, the chemistry is sort of there. The rate constants help to define how quickly the reaction can occur. So if you're in a system that's ozone limited, any change in ozone will affect how quickly products are formed. And so that's where the kinetics information can give you that's how the modelers can use the kinetics information to predict what products are formed based on the ozone, what the yields of those products would be based on the ozone concentration.
Great. The next question I have here is, in addition to the impact of some of these typical indoor reactants that you covered, like the OH radical, NO3, ozone, have you looked at the impact of light on indoor chemistry?
That is a very, very good question, and that has just now starting to be investigated. And it's a little surprising to think that people haven't really looked at that in great detail. But it was always assumed early on that light didn't play much of a role in the chemistry. But there are some recent research, some models out of Penn State that are showing that indoor lights, certain type of lights, can create certain spectra that could impact secondary chemistry as the products are formed. So that's an evolving research area. And of course, what's also interesting too is light mixtures are changing now too. So as we shift from incandescent obviously to LEDs, the spectrums are changing as well as the intensity, and so that's going to impact some of that chemistry as well. So that, again, that's a very good question. We also have the difference between occupational sites and lighting needs there versus the home environment. I think, if I remember correctly, the Nazaroff and Weschler paper talks about light in that paper. So, but again, that's an evolving area of research.
Great, thank you. It sounds like there's a lot more to explore there. Our next question is referring back to I think it's slide 36, about the particle composition from indoor and outdoor particles.
Yeah.
The participant asks, the particle composition for outdoor HOA was 8%, while the indoor HOA was 19%. Is that common when comparing outdoor and indoor particle composition, i.e., does HOA tend to be higher in indoor particles versus outdoor particles?
Well, I think one of the things that's important to think about from the paper is to show that the HOA increase, they hypothesized in the paper in part because of reaction products that were present in the indoor environment that were of sufficient low vapor pressure that they partitioned, especially to the new particles as they were coming into the indoor environment. So that's where they think the HOA increase came from, is the low vapor pressure products that I talked about earlier that were already present in the in the gas phase, ended up partitioning onto the particulate phase.
Okay. Let's see. Another question, have you run any scenario-based experiments to determine the emissions of specific indoor activities?
Yes, there are some publications that are, I think, in the works now on, I think, not only cleaning, but there's some other activities like exercise or cooking, especially, and this is where the home chem project has been so interesting, because they looked at some very kind of routine everyday activities and really tried to break them down into their chemical impacts, and so that's again, that website for the home chem can give you a lot of insight in current activities and their relationship to chemistry. And again, Nina Notman's paper on occupant emissions is important as well.
Great. And I think, if I remember correctly, I think home chem did do some scenarios with different cleaning scenarios, right?
Right.
Okay, that'd be a good source of information for that as well. Our next question here, you talked a little bit about the health risks and the various chemicals and kind of some of the mixtures. This participant asked, in real life, people are exposed to multiple chemicals together. How are those risks measured in the context of other chemicals or other exposures?
Again, that's a very good question. And right now, the look of or assessing a risk of a mixture is still being investigated, because it's not just the chemistry that you have to think about. You've got to think about the receptor, the human or the biological system, and those complexities make assessing risk a little bit more challenging. But there is great interest in, because we've got such improved computer models and the chemistry understanding is increasing all the time, that these tools are being used to at least give a better ballpark of risk, rather than trying to look at individual chemicals and sort of either adding up all the risks or blending them together, because sometimes there can be combinatorial effects that can either be worse or better. And so that complexity is just now really being addressed. And so that's a very timely question, and one that I'm very interested in as well.
Definitely. Another question here. So I think we have several folks who are not necessarily chemists in the audience. So this person specifically asked, would it be correct to say that things like room deodorizers can combine with ozone or OH radicals to add to the levels of VOCs indoors? And I would kind of add to that, just if you could summarize some of the takeaways that folks can take home from this presentation today.
Yes, I mean, I think that's, I'm not trying to alarm anyone because we do have products in our home and we continue to use products, and you know, cleaning is a beneficial activity. You know, so it's a little, it can be a little challenging to try to turn this into, you know, to see if there's a binary yes no answer. And you know, one of the observations from some of the models were to be, you know, if it's possible to clean on low ozone days. You know, if you're going to use a fragrance cleaner, simply because you reduce the chances of that ozone chemistry happening. So, you know, I think, like a lot of things, the key is to try to use some of these products in moderation, to try to use other products that maybe are not as fragranced. You know, ventilation is an option as well, but there's, again, this, like a lot of nonlinear processes, ventilation there's a trade-off, because if you increase the ventilation to remove chemicals, you're also bringing more outside air in, and so you can increase your particle as well as your the ozone concentration. So the answer is it's not very clear cut, but there are a couple of changes that one can make, again, using reduced fragrance products, as well as you know, if you can, to try to do any cleaning on lower ozone days, so that you reduce that reaction.
Great. Definitely a complex system. All right. Another question about a specific species that you mentioned. You talked about 4-OPA.
Yes.
And that a 65 ppm concentration is pretty high. What's the typical concentration of 4-OPA indoors? And are there indoor sources, or does it come from outdoors?
Well, it can come from, well, let me answer the first question first. If you remember from my field sample slide, I had like 75 parts per trillion. So 4-OPA is, you know, we're talking several orders of magnitude to get to parts per million. So that's, the concentration is not all that high, but it's certainly indicative of chemical oxidation occurring indoors. Now, again, those were gas phase species. I wasn't looking at particulate matter, so there could be other dicarbonyl species present in particulate matter. And the indoor sources would be the terpenes, some of the terpenes, depending on the reaction mechanism, once they react with ozone. And another indicator is that 4-oxopentanal is a secondary reaction product of squalene oxidation. So it's another indicator of occupant influence on indoor chemistry. So what happens is that squalene, as it's oxidized, makes a compound that's a little bit larger, called we call it 6-MHO, but it's basically a single carbonyl with a carbon-carbon double bond, and then ozone reacts with that to make the 4-oxopentanal product.
Great, thank you. We had a question from one of the participants. At one time, we were taught that ozone was a very short-lived chemical indoors. Based on what you've presented, is that still true, or does it not matter that the ozone can react rapidly enough that we should be more concerned with the chemical compounds that are formed in the reaction?
Well, you know, ozone ozone's not as reactive as the OH radical or the NO3 radical. And the fact that it's measured indoors and people can relate the concentration indoors to the outdoor concentration certainly suggests that ozone may be lost on a few surfaces on the way in, but it is transported indoors and present indoors. So it's, as an oxidant, it's really the one that basically starts the process. So it is reactive, but in the indoor environment, it's present in high enough concentrations to initiate some reactions.
Great, thank you. Let's see. Another question was, are you working with modelers to help determine relevant compound concentrations to expose biological systems when looking for inflammatory effects?
That's also a very good question. I do have some collaborators that are modelers, and the approach that I would be taking would be to try to see what the model suggests are the sort of ultimate products that are formed, and then build an exposure mixture, if you will, if possible, of the compounds that are relevant into the chamber or on particulate matter, and then to deliver those to the biological system. As I mentioned earlier, there's still some great interest in looking at cellular exposure and trying to interpret health effects from those responses. So the question about modeling is obviously a good one, and it's one that's efficient. But again, models certainly can make some assumptions that we need to make sure are either correct assumptions or we can adjust them. So there's still a lot of growth in the modeling world as well.
Definitely. We've had a few questions that I'll try to combine here about different types of air cleaners, for example, UV sterilizers or ionizing air cleaners or hydroxyl generators, and how those impact indoor chemistry. Do you have any thoughts on that?
There have been several publications. This was a very contentious issue, as you can imagine, a few years ago. And there have been several publications looking at the impact of these devices on indoor chemistry. And from my memory, I don't think they were glowing reports, so that there were some challenges at the introduction of these systems into the indoor environment, and the question of whether they were either helpful or harmful. So some of that was put into question. Those, again, there's a lot of literature out there on those topics, and that should give at least the people that ask that question some insight onto whether they should choose those or not.
Yes, definitely. So one participant asked, based on what you've presented, should we be looking for ways to reduce ozone or OH radicals in the indoor environment, or what are some of the ways we could reduce some of those species?
Again, that's another excellent question, and there have been experiments going so far as trying to put different coatings on walls to react with ozone and pull it out of the system. I think one of the challenges is since ozone's brought in from the outdoors typically, is to try to reduce it before, or eliminate it before it even gets into the indoor environment. And that can be a challenge because of the filtering systems that could be used. That can be a big draw on both the system as far as energy goes and power usage. So that's one of the challenges with these large commercial buildings is trying to reduce the power consumption and save costs there. And so that, the ability to prevent ozone from getting into the system could be an important step. I think the other challenge too is that, again, I think what we'll see too, like the outdoor environment, it's not a linear process. So just removing one component may open up chemistries for other things. So it's something that the experiments, you just have to try to figure out what these interrelationships are, and that's in a mixed environment that can be a little challenging.
Great. Let's see, I think we've got time for about one more question here. We have a couple of questions related to ventilation. One kind of related to ventilation controlled by CO2 measurements, which suggests ventilation when more people are around in a building. And cleaning, on the other hand, is typically done when an office building, for example, is empty. Would the control with CO2 for the ventilation system be sufficient in that case, or would it be would some more controls be needed to reduce some of the byproducts of cleaning there?
That's, again, these are interesting questions, and unfortunately, since my experience is not in ventilation, I can't really answer them definitively. But I do like the idea of the CO2 measurement as sort of a feedback loop to change the ventilation parameters in a space. Again, that requires more manipulation or software or sensors to try to detect all of that information, but that may be pretty useful. I think, again, one of the things that there may be some insights, and I haven't read all the papers, but from the home chem experiments, there could be some insights there on that interrelationship between cleaning and time of day, as well as cleaning and occupancy as well. And I think one of the challenges too is this is not, you don't just clean and that's kind of the end of it. You can have some buildup of things over time, and so you have to be just cognizant of that as well.
Absolutely. I think that is all the time we have for questions. We're running up on 2:30 here. Thanks again, Ray, for joining us and for this excellent presentation, excellent Q&A session. It's been fantastic to have you here with us today. And thank you also to those of you who joined us online for submitting your questions. For announcements of future IAQ Science webinars, you can go to epa.gov/IAQ for more information both on our webinars and on indoor air quality. So thank you again for attending today.
Thank you very much.
All right, and with that, we'll close the webinar. Thanks and have a great day, everyone.